It's a moment of extraordinary joy to have the keynote delivered by Professor M S Valiathan, National Professor, who has earned a place in the Hall of Fame as one of the most distinguished Indians in contemporary history. Introducing Professor Valiathan to an audience is both a pleasure and a challenge. Pleasure because his has been an inspiring life with many lessons for all. Challenge because his achievements are far too many to be recounted in a few minutes. Professor Valiathan was a cardiac surgeon whose grand vision led to the creation in the 1970s of the Sri Chitra Institute for Medical Sciences and Technology, Trivandrum, an institute of national importance under the Ministry of Science and Technology, Government of India. The institute, under his leadership, developed the first indigenous heart valve that has today been implanted in more than 150,000 patients in India and abroad. Professor Valiathan was awarded the Padma Vibhushan in 2005 for his pioneering contributions to health technology in India. An elected fellow of all the science, medical and engineering academies, he is a past president of the International Science Academy. He is a recipient of many international and national awards for science, technology, and education. In 1999, the French government honored him with the title of Chevalier in the Order of Academic Arms. Professor Valiathan received the Dr. Samuel P. Asper International Award from the Johns Hopkins University Medical School in 2009 for his contributions to international medical education. He is a Hungarian professor of the Royal College of Surgeons of England. Other awards received by him include, among many others, the R.D. Birla Award, O.P. Bhasan Award, Jawaharlal Nehru Award, the Dhanantari Prize, Aryabhatta Medal, Basanti Devi Amarchand Prize, and the J.C. Bose Medal. More recently, his foray into Ayurveda culminated in his authoring 5 books on the subject, including the 3 on Charaka, Sushruta, and Vakmata. We request you, sir, to deliver the keynote address online.
Thank you. When Dr. Shivakumar asked me to give this keynote, I was very reluctant because I felt very insecure because I have spent a lifetime in cardiac surgery and I had no knowledge of biotechnology except what I read in news items. To come to a meeting where experts, scientists who have spent a lifetime in biotechnology to come and give a keynote, I was not very confident. But I came here because I cannot say no to Dr. Shivakumar, a valued colleague of mine. I must tell you how I got into this, because towards the end of my 30-year career in surgery, when I was in Chitra Institute, a thought started bothering me, because in the operating room where I spent a lot of time every day, all that I saw there, all that I heard there, the human beings, the patient, the surgeon, the assistants, nurses, they were the only Indian part of the scene. All the rest, the concept of open heart surgery itself, the drugs that we use, the machines that we use, life-saving equipment that we use, all these things had originated abroad. Nothing could be claimed to be Indian in origin. Now, this was a disturbing thought. And later on, thinking about it, it occurred to me that all the rest of advanced medicine, the situation is no different. Whether you take orthopedic surgery or anything else, or medicine, dialysis of patients, you take anything. Now, this was a very disturbing thought. Western medicine had come to India in the 19th century, middle of the 19th century. And all these years, there was not a single disease in India, the cause of which had been discovered by an Indian. Work was done here, like Ronald Ross, vector transmission of malaria, very great discovery, but it was done by an English, by an Indian. Plague vaccine was developed in India for the first time, not by an Indian, it was done by a Russian Jew, Afghan. I can give you any number of examples. I couldn't find any exception. I was taken back to a statement made by Nobel laureate Shockley in 1960s, he created a huge controversy at that time. He said the path-breaking discoveries in science, they can only be made by white people. Others are incapable of doing this. This created a very big controversy at that time, but he stood his ground. I started wondering, maybe he's right. There is an element of truth in that. But immediately, a comforting thought came to me that modern medicine, Western medicine, came only in the 19th century. But we had a system of medicine in practice for many hundreds of years earlier. Even in Buddha's time, they had a practice of medicine here. They didn't call it Ayurveda, they call it Chikitsa. Charaka goes back to 2000 years. Sushruta is even earlier. So is there anything there we could claim as original thinking, which did not exist anywhere else in the world? That is how I ended up in studying these Ayurvedic texts from the Acharya. And there I began to see, like, for example, Sushruta's own description of surgical diseases, of the operative techniques that he used, the instruments that he invented, described in great detail. All these are there. These were not borrowed, they were original. There is some disturbance in this. There is some disturbance. Can you hear me?
Yes, sir, you're audible.
Now the interaction of Veda with Western medicine, that started only in the 16th century. When the Europeans came here, the Portuguese came, there was Garcia de Horta, a Portuguese physician, who came here and set up practice in Goa. He started practicing medicine, and there were not enough European physicians to look after them. There were not enough European physicians. They had to depend on native physicians. Many of the diseases were unfamiliar. They did not know how to treat snake bite or dysentery. They were not familiar with them. So they had to depend on native physicians, Vaidyas in Goa, or some variant of Ayurveda, folk medicine, whatever you call it. They had to depend on them, depend on some of these native preparations, including the governor of Goa at that time. That was the first interaction. And he wrote a very classic book, Colloquies on the Simples and Drugs of India, which became a bestseller in those days, translated into many European languages, of Garcia de Horta. That was the beginning. Great emphasis on medicinal plants, preparations of medicinal plants, looking at it from the Western point of view. Very good description of diseases, which we did not have. For example, cholera, many of them which he describes, they are not described in that much detail in Ayurvedic texts. But next came 17th century. This was followed by our own Hortus Malabaricus. You know that story very well. That was done right here in Kerala. More than 700 plants, classified, described in great detail, beautiful illustrations. And the medical applications are also given there. There was an Ayurvedic physician, Gitti Achyutan, who was giving that information. That was in the 17th century. Again, taxonomy was reigning supreme. This continued for the next 200 years all over India, including Chennai, Roxborough in Bengal, who was greatly admired by J.C. Bose. He even wrote a book, Hortus Bengalensis. It never got the popularity of Hortus Malabaricus. But that all through taxonomy, looking for medicinal plants and possible cure for tropical diseases. But this went on, and 20th century only, a new development came. That was Ramnath Chopra, Calcutta School of Tropical Medicine. He introduced pharmacology for the first time, that these plant extracts could be made, compounds could be isolated, purified, effects tried in experimental animals. That whole culture of pharmacology came with Ramnath Chopra. In fact, he is rightly acclaimed as the father of pharmacology in India. Now, this was followed by natural products chemistry. It was a natural development, very many great names. like Ashima Chatterjee in Calcutta, Govindacharya in Madras, many of you know. And that food, natural products chemistry, India became a leader in the world. Large number of plants were classified scientifically, a large number of papers published, a very large database. Even today, it is useful for people in phytopharmacology. But this went on. But then, middle of the 20th century, as you all know, the double helix came, and that changed the way we look at life itself, because we could understand genomics, how genomics could explain many of the biological phenomena. It was natural that Ayurveda would be viewed not only through taxonomy of pharmacology, of natural products, chemistry, but also it should be viewed through the modern science that we have, molecular biology. Now, this is what made us realize, for example, the Ayurveda classifies human beings into three basic types. They are actually constitutional types, Vata, Pitta, Kapha. This is all done on clinical determination. But is there a genomic basis for this? First, we had the power to do this. We had the techniques. We could do the genotyping of these. Is there something which differentiates these three types? That kind of basic questions. These could not be answered earlier because that genomics didn't exist. When I was a medical student, for example, there was no way this could be determined. Nobody even thought about this. But this changed. So that is where we thought there should be a new way of looking at Ayurveda, Ayurvedic biology, using the new tools of biology. And the Indian Academy of Sciences published a vision document in 2006 towards Ayurvedic biology. That was the beginning of a new science. And we have subsequently, large number of papers have been published, a lot of work has been done, and more people. There are actually courses in Ayurvedic biology being offered by 3 or 4 universities in India. Now we had, this is where it began in 2006, and the first 10 years, if you look at broadly what was done by different groups in India, I just want to give a few examples for you, what this means. Take, for example, this Vata, Pitta, Kapha, that constitutional types of human beings. It is extremely important because in Ayurveda, the predisposition to diseases, that depends on these types. In other words, the Vata type of people, they are prone to get certain diseases. Pitta type, they are prone to get some other diseases. This is well described. Now, that is very important for the incidence of diseases, for diagnosis also. And secondly, the therapeutic response, that is also different. If you give the same treatment to these three types, the response need not be the same. It will differ. Now that response to treatment, so these two are extremely important, dependent on these types. But is there a genomic basis for this? Or is it only simple clinical determination, looking at the height, looking at the kinkiness of the hair, roughness of the skin, their behavioral pattern? So what is Ayurvedic physician looks at in determining these constitutional types is essentially physical criteria, mental criteria. Some people get easily scared, other people are not scared, like that. All these are considered, and behavioral types. All these three characteristics are determined. That is how they decide this is Vata, Pitta, Kapha. Now, is there a genomic basis for this? It had never been established. And now this is the first project we did, the major project, constitutional types, Vata, Pitta, Kapha. Is there a genomic basis? That was the central question. This is a big project funded by the Department of Science and Technology, involving several centers. The main centers involved, one was our own Manipal Life Science Center, Indian Institute of Science, CCMB, Hyderabad, and the University of Pune, Biotechnology. These are all the centers. Ayurvedic centers were involved, one in FRLHT in Bangalore and the ST College in Udupi. They were the Ayurvedic centers contributing to this. Now here the samples, about 3,000 subjects between 20 and 40, healthy young people. And the dissemination of this Vata Pitta. This was done by the Ayurvedic people from volunteers. Now, once they decide Vata Pitta Kapha, there could be inter-observer variations. So we wanted another confirmation of this. And there had been a software developed called IUSoft in Pune. They had never actually used it by simply giving scores for the various traits. If there are 100 traits for Vata, each trait it is not the same. So Ayurvedic experts, a committee went through this and allotted scores for this. So we had numbers. So Vata numbers are predominating in one group, Pitta in another. So we had a way of classifying them based on this, the IUSoft from Pune. Now that classification, we were actually concerned initially, a clinical determination done by Ayurvedic physicians, will it have any correlation with this IUSoft determination, which had never been done before? But we were surprised that there was a correlation of over 80%. So there is some science in this clinical determination. It is not just a guessing. So that was the basis for selecting out of these 3,000, some 280 we could classify that predominant Vata, predominant means more than 60% of the score belongs to that. That's how we selected some 280. And the SNPs, that was the marker, genetic marker that we are using, 1 million were chosen. And there were 52 of them, what I remember. That was enough according to the statisticians to give a classification into these 3 types without taking any aberrant factors, excluding all that. Now, this study which has been published in Nature Reports in 2015, which received a tremendous response, that showed very clearly that there is a genomic basis for this classification that had been used in Ayurveda. That was the first major project that was done. That was a concept in Ayurveda. And subsequently, this DNA methylation was used as a marker by Satyamurthy's group, Journal of Translation Medicine. They also confirmed this. There have been other studies, I won't go into that. But that is an example of Ayurvedic biology confirming or giving a scientific basis for an ancient concept. Another type of study that we do in this, that is looking at various procedures. Is there a mechanistic basis for this? For example, we have a category of drugs called rasayanas in Ayurveda. Rasayanas are actually to improve the health or resistance to disease. All these are comprehended by this term. It is not to treat a disease. It may have a collateral effect on treatment, but basically it is to strengthen the body and make it protected from diseases. Now, is there any way we can demonstrate this effect, positive effect on the system? We had Dr. Kalluri Subbarao, some of you may know about him. He was primarily interested in aging research in Hyderabad, University of Hyderabad. For some 30 years, he had been working on this using DNA chain breaks of neurons and astrocytes in rats. So the various types of assay systems he had developed. He's a very well-known scientist. So I asked him, he's a friend of mine. So I asked him one day, Dr. Subbarao, these tonics, there is one very commonly used called Amalaki Rasayana or Nellika, widely used. Charaka describes it, all the acharyas describe. Now I asked Dr. Subbarao, do you think we can test the effect of this on rats? Rat versus model for experiments. He gladly agreed. He said there is no problem at all. So Kottakkal made this Amalaki Rasayana for us as per the original description in the text. And this was given to the rats, his experimental animals. His method was he would study the brain, neurons and astrocytes at 3 months, 9 months, and 15 months. They usually live around 2 years. So this was the time intervals at which the animals were sacrificed and the brain studied. And in the control animals, which were not pre-treated with any medicine, by the time they reach the 15 months, the DNA disruption is double and single chain breaks were extremely high. This was the standard we had established over many years. But the animals which were given pre-treated with Amalaki Rasayana. This was very significantly shown. Even a lay person can look at it and see it is very different. So that was published in one of their aging journals, very prominent journal. It attracted a lot of attention. So that was in the rat, this tonic effect on the brain, that is the stability of the genome is protected by this. There was a very clear evidence of that. This paper was seen by Dr. Lakhotia in Banaras, as many of you will know, in Drosophila genetics. He's an authority. He had seen this paper, so he asked me, do you think I can try this in my fruit flies? We don't know whether fruit flies will take Amalaki Rasayana, but there is no harm in trying. So the same Amalaki Rasayana was supplied from Portugal. He repeated these experiments. And that came out as a revelation for him and to many of us. This had a very positive effect. Their lifespan was increased significantly. A number of biomarkers, for example, starvation tolerance, heat tolerance, fecundity in terms of eggs being laid, heat shock proteins production, number of parameters. All these were enhanced. And this was biomarkers, very dramatic enhancement. This was published in PLOS One. Again, a lot of very positive response for this paper. And subsequently, the Lakhotia's department, there were 3-4 others who joined him because they were excited by this. There were a large number of papers on this. In fact, another paper which they did was on the genetically modified Drosophila. He had a colony of Drosophila, which Huntington's chorea and Parkinson's. Now, these degenerative changes in the brain, they were inhibited by giving this. This was not even expected. I'm quoting from published papers only. So that was another in the fruit fly model. First we had the rat model, fruit fly model. What about the human model? There it is very difficult to do these experiments. You cannot do it. But one of the experiments done was in Mumbai. Dr. Urmila Thatte, who is a very well-known clinical pharmacologist in KEM Hospital, and Dr. Chiplunker, who is the head of ACTRAC. She's a very distinguished immunologist. They were the two investigators. Here, the human study, it was Panchakarma which was being tested. In Panchakarma, I will not go into the hypothesis. Incidentally, in Ayurveda, whether it is Rasayana or Panchakarma, these procedures which are done, it is not simply, it was done in the past, so we repeat it because the past experience was good, so our experience should also be good. That's not the way it is done. There is always a hypothesis in this. In the Rasayanas, when you give, how do they improve the tissue conditions? How do they give protection? We may or may not agree with their hypothesis, but they always have a hypothesis, very scientific. And similarly, in Panchakarma also, there is a way body collects metabolic products which are sometimes harmful, catabolism. Now, how to eliminate it from the body? Because body is a closed compartment lined by the mucous membrane within the gut, starting from mouth to the anal canal. That whole gut is lined by mucous membrane, which guards the internal frontier of the body. Skin outside, that guards the frontier from outside. In between is a closed compartment. You have no direct access to it unless you inject. You have to break that. Now, how do you, if these toxic substances, they call it toxic, they're collecting, how do you eliminate this closed space? Now, Panchakarma was their method of doing this. I won't go into the details, but they had given a lot of thought, much of it imagination, but imagination is the basis of hypothesis. Now this particular paper, the work that they did was on obesity. Obesity, in my student days, it was considered a lot of fat collecting and sitting there, occupying a lot of space. That was the way we looked at it. But today, obesity is no longer like that. Obesity is a disease. This is not inactive substance collecting there. It is actually working. It is producing. All these pro-inflammatory cytokines are being produced. And they not only produce this, they have ill effects, all this insulin resistance. That is actually promoted by these cytokines. The method which they followed, Ayurvedic treatment is using in Panchakarma, the basis is enemas. Enema is not Panchakarma. Panchakarma consists of first a lubricant substance, fatty substance is taken, and then they are subjected to Svedana or sweating in a Turkish chamber. A lot of sweating takes place. Then only they give this vomiting, toxic substances collecting in the upper half of the body. They are given an emetic, so they are made to vomit. If it is the lower half of the body, they give a purgation enema. So in obesity, especially with pot belly, this enema is the major form of treatment, Panchakarma. Now, this was the experiment they did. Some 30 subjects, very obese, more than 30 kilograms per square meter. Now, that study was published in the Indian Journal of Medical Research. There you will see, once they start doing this Panchakarma, after the first treatment itself, these pro-inflammatory cytokines level originally high, that begins to come down. Middle of the treatment, it is 3 weeks treatment. Middle of the treatment is further coming down. And 90 days after the treatment was concluded, still these levels remain low. Now, this is a very important finding. Nobody, Dr. Chiklungar told me initially, how do you think, how can enema produce an immunologic effect? How is it feasible? So she was a skeptic. But when she found this, she herself found it very difficult to explain this, how this happens. We still don't know that. But that is an example in human beings being shown how an ancient method is working, and also some idea about the way it works. So I feel that many of these, we have only scratched the surface. There are a whole lot of things in Ayurveda. For example, even Atharva Veda, which is the forerunner of Ayurveda, there is a mention, animals when they are sick, dogs, for example, pigs, they don't normally eat grass. But when they are sick, you will find them sniffing, going and eating some plants selectively. What's the basis for this? How do they choose? When I was reading this, I found a lot of research had already been done on this subject in Africa. And there, monkeys, they have done extensive work on that. Again, they go and eat. So they have very exquisite sensory, taste, and sensation. Receptors which are extraordinarily sensitive. Now that whole area, taste and smell. I found in the United States there are institutes of taste and smell. We don't know in what all ways this could become useful in treatment of patients. But it's an area which should be explored. We even called for proposals on this. We couldn't get in the task force in Ayurvedic biology. So I'm just telling you that we have only scratched the surface. There are many, many other areas, especially in 2 major areas, the degenerative diseases of the central nervous system, where it has a lot to offer. And the second is locomotor, musculoskeletal system. These 2, I don't think, for example, emergency medicine, there is not much Ayurveda can offer there. Many surgical conditions it cannot offer. But these are areas with the non-communicable diseases rising in proportion. Today in Kerala, for example, the largest cause of mortality and morbidity will be non-communicable diseases, not infectious diseases anymore. Now, this is precisely the area where Ayurveda has a lot to offer. And so I hope this will only succeed if there are good Ayurvedic physicians interested in investigation and good scientists, biologists, who are very familiar with the modern techniques, who are also interested in this. In the last, I was there in 10 years, I was the chairman of the task force in Ayurvedic biology. One of the things which distressed me was there were no research proposals from Ayurvedic colleges. Even though in our project in the task force, it was compulsory that the projects should be jointly written by Ayurvedic physicians and scientists. So there would be a name, but they were not really intellectually participating in this. But subsequently, we never got any proposal from Ayurvedic institutions. And secondly, even more distressing for me was if there was a positive lead in one study. For example, there is clearly a genomic basis for these prakritis, Vata, Pitta, Kapha. There is no, that lead is not used as a nucleus for starting a new project. This way, that science will never progress. So we found that as soon as the grant stopped, all the work stopped, they will change to something else. So that way, you cannot build up as science. So these are some of the problems. So I hope the brightest scientists, biotechnologists, biologists, they will all take interest in our heritage. I can assure you there will be very, very important findings. When somebody, for example, the genomic basis of diurnal rhythm, that received Nobel Prize, everybody was talking about. Ayurveda also has this rhythm, they have described it. That because the people who got the Nobel Prize, when you take a basic thing like the smell, driving an animal to find a medicine, that kind of a natural observation, which was made in Vedic times, if you want to take it to the Nobel Prize level, it needs something like 10 years of work, sustained focus, and tenacity. Otherwise, you cannot really achieve much in science. I mean, I'm talking about basic science. So this is all I would like to say. I hope there will be some more interest created by a webinar like this. So many young scientists will take some of these problems interesting and carry it to great heights. I would stop and thank you very much for this opportunity you gave me to address you today. Thank you.